Hello, my name is Pedro Oliveira. I'm a senior scientist at Mount Sinai School of Medicine, New York, and I'd like to thank BBM for this opportunity to talk a bit about conserved DNA methyltransferases and their role as keystones of epigenetic regulation in bacteria. So we know that in this age of post-genomics, the majority of research has focused on decoding the sequences of the four canonical DNA bases, adenine, cytosine, guanine, and thymine, and making sense of its meaning. But on top of this information, there is an extra layer of epigenetic data encoded as covalent modifications to these canonical bases. So these modified bases effectively expand the DNA alphabet beyond 4 nucleotides in a process mediated by DNA methyltransferases. In humans, for example, the methylation at the 5th position of cytosine within a CpG context is an important epigenetic mark that can affect embryonic development or differentiation, for example. Bacterial genomes, on the other hand, are also methylated, but with just a few exceptions, namely defense against DNA invasion or involvement in DNA mismatch repair, in cell cycle control, and transcription regulation. Rather little is known on the functions of the multiple DNA methyltransferases and corresponding epigenomes that are out there. So we are particularly interested in better understanding the roles of DNA methyltransferases in bacteria, particularly conserved ones, which by definition are those present in all or almost all of the genomes of a phylogenetically coherent group, also known as persistent or core. And for this reason, they are more likely to play key functional roles. Examples of well-characterized ones include the Escherichia coli DAM enzyme and the Colobacter crescentus CCRM. So whether DAM and CCRM are merely outliers or actually representatives of a broader set of conserved methyltransferases is currently not clear. So to gain a clearer view of the actual landscape of conserved DNA methyltransferases in the bacterial kingdom, we started by performing a large-scale screening of a set of 139 bacterial species, those for which at least 10 complete genomes were available in GeneBank, which are here colored by phyla. So the total data set analyzed corresponds to close to 6,000 complete genomes. The heat map that you see here corresponds to the density of methyltransferases per genome per megabase for each species. And the blue bar plots correspond to the percentage of most abundant methyltransferase detected in each species per type. So we consider a methyltransferase to be conserved if it was present in at least 80% of all genomes of a species. Surprisingly, we found a total of 144 conserved methyltransferases. 52% of the species analyzed actually contain conserved methyltransferases. So this means that we don't have only DAM or CCRM. These results show that we have actually a huge reservoir of previously unexplored conserved methyltransferases awaiting to be studied. So as a proof of concept of the former large-scale analysis that I just showed you, we selected one of such conserved DNA methyltransferases, a previously not studied one from the human pathogen Clostridioides difficile, and performed the first comprehensive analysis of its DNA metylome using third-generation long-read sequencing technologies, namely smart sequencing and comparative epigenomics. We found a conserved solitary methyltransferase whose inactivation impacted sporulation, which is the main route of transmission to a human host, both in vitro and in vivo. And we also found evidence for epigenetic regulation associated with cell length, biofilm formation, and host colonization. And here on the left, you can see an overall depiction of the epigenome of C. difficile, as well as the complete landscape of target methylation sites detected by smart sequencing. So as a take-home message, I would like to highlight the fact that we found multiple unexplored conserved DNA methyltransferases in bacteria, which holds tremendous interest for future epigenomic studies. Also, the fact that third-generation sequencing technologies, such as smart sequencing, have been catalysts for the bacterial epigenomics field. And as a proof of concept of the smart sequencing potential and also of our large-scale analysis, we found that one of these previously unexplored conserved methyltransferases in C. difficile has a role in sporulation, biofilm formation, and colonization, opening doors to the development of epigenetic inhibitors. So my acknowledgements go to multiple labs, not only from Mount Sinai, but also from Tufts, University of North Carolina, the Pathogen Surveillance Program at Mount Sinai, Pasteur Institute, New England Biolabs, and of course the multiple sources of funding. Thank you very much.